Welcome to this video on the fundamental unit of life. We will be covering various topics including the cytoplasm and different cell organelles, such as mitochondria, plastids, the endoplasmic reticulum, the Golgi complex, ribosomes, vacuoles, lysosomes, centrosomes, and peroxisomes. The cytoplasm is the fluid that fills up cells. It was discovered in 1835 and is now considered similar to a glass-forming liquid. In eukaryotic cells, it includes all cell contents except the nucleus. In prokaryotic cells, the cytoplasm holds the genetic material. Together with the nucleus, it is called protoplasm. The cytoplasm is composed of cytosol, cell organelles, and the cytoskeleton. The cytosol is a rich broth of molecules where many metabolic reactions, like protein synthesis, occur. Cell organelles are specialized parts for specific metabolic processes, while the cytoskeleton is a network of fibers that supports the cell and gives it shape. The cytoplasm has several functions, including the transport of materials, maintaining cell shape and structure, protection, storage, and serving as the home of metabolic processes. We will now discuss the various cell organelles, including mitochondria, plastids, endoplasmic reticulum, Golgi complex, ribosomes, vacuoles, lysosomes, centrosomes, and peroxisomes. Mitochondria are rod-shaped, double-membranous organelles. The inner membrane folds into cristae. They contain their own DNA and ribosomes, classifying them as semi-autonomous organelles. Known as the powerhouse of the cell, their main function is producing ATP, the energy currency for cells. Plastids are double-membranous organelles found in plant cells and are also semi-autonomous. They are classified into chromoplasts, colored, chloroplasts, green, and leucoplasts, colorless. Functions include making food through photosynthesis, earning them the nickname kitchen of the cell, and aiding in pollination. Plastids have various types, each with specific functions, such as chromoplasts for pigment synthesis and storage, chloroplasts for photosynthesis, and leucoplasts for the synthesis of fatty acids and amino acids. The chloroplast structure includes outer and inner membranes with an intermembrane space. The inner matrix, or stroma, contains grana, which are stacks of thylakoids. The light reactions of photosynthesis occur in the thylakoids, while the dark reactions take place in the stroma. The endoplasmic reticulum is a system of membranes attached to the nucleus. The rough endoplasmic reticulum, or RER, has ribosomes and is involved in protein synthesis. The smooth endoplasmic reticulum, or SER, lacks ribosomes and is involved in detoxification, fat, steroid, and cholesterol synthesis, and supports the cell framework. Membrane biogenesis is the process of cell membrane development, driven by the endoplasmic reticulum, using proteins and lipids. The Golgi complex is found in eukaryotic cells, composed of interconnected cisternae with cis and trans faces. It forms vesicles for post-processing cellular secretions. Its functions include secretion, protein modification, cell membrane formation by processing lipids, and forming lysosomal proteins. Ribosomes are the smallest organelles, dense, spherical, and granular RNP particles made of large and small subunits. They attach to mRNA to form polyribosomes. They are known as the quote-quote protein factories of the cell, as they are crucial for protein synthesis. Vacuoles are temporary storehouses surrounded by a membrane called the tonoplast, and they help maintain osmotic pressure. Lysosomes, also known as suicidal bags or scavengers, are tiny spherical sacs with powerful digestive enzymes. Their function is to remove worn-out organelles and digest foreign particles. Centrosomes consist of two sets of cylindrical, rod-like centrioles, each made of microtubules. Their main function is to help in cell division by forming spindles for chromosome attachment. Peroxisomes are small, membrane-bound organelles in eukaryotic cells, similar to lysosomes but with distinct content and function. They consist of a single lipid bilayer membrane, a matrix containing enzymes, and a crystalline core. Functions include lipid, amino acid metabolism, detoxification, and the biosynthesis of lipids.